Good afternoon, everyone. Thank you very much to the organizer for this opportunity to present my work today. My name is Maria Maoun, and I am a postdoc at Washington State University. For today's talk, I am going to share with you some of my work on SuperSoft Wheat that was done in collaboration with Dr. Aaron Carter, Brian Ward, and Craig Morris. This is the outline of my talk. I will start by giving a brief introduction on grain hardness and the SuperSoft kernel texture and their importance in wheat quality, and then on our work to determine the genetic factors underlying the SuperSoft grain texture, and our ongoing work to develop molecular markers to assist breeding for SuperSoft wheat. Grain hardness is a main determinant of wheat milling and baking quality. It is measured using different methods, but the most recent and common method is based on the single kernel characterization system. This is a system that crushes individual kernels and then uses an algorithm that accounts for kernel size, weight, and moisture to generate a unitless hardness index. The higher the hardness index, the harder are the kernels. Based on grain hardness, wheat can be classified into different classes: soft, intermediate, hard, and very hard. Harder kernels, they require more power to mill, and they produce flour with larger particle size, higher damage, starch damage, and higher water absorption compared to flour from soft wheat. So the products from each of these wheat classes are different. For example, soft wheat is mainly used for making food products like cakes and cookies, while hard wheat is used for making bread, and very hard or durum wheat is used for making pasta. We know that the pyrandoline genes that are located at the hardness locus on the short arm of chromosome 5D are the main genetic factors that can distinguish between soft and the hard wheat. However, within each group, we see a quantitative variation in grain hardness indices, and this is an indication of the presence of minor genes that can be involved. In this figure, I am showing the distribution of grain hardness in normal, in soft wheat, where you can see this quantitative variation in grain hardness. This is a graph taken from a study by Morris and others, where they characterize a unique grain texture called the SuperSoft that is known by lower than usual grain hardness values compared to the normal soft wheat, where the ranges are usually between 20 and 30s. So for SuperSoft wheat, we can see grain hardness values that can even go to negative values, as shown in this example of -2 and even lower. SuperSoft kernel texture are associated with superior flour quality, and the reduction in grain hardness was associated with higher break flour yield, lower water absorption, larger cookies diameter, and increase in sponge cake volumes. And we did not see any indication of negative effects on the commercial quality of soft wheat. So SuperSoft wheat actually represents or provides an opportunity that can to improve wheat processing and flour quality. However, up to this point, we didn't know the genetic basis behind the SuperSoft kernel phenotype in wheat. We know that they are independent, or this kernel texture is independent of the pyrandoline genes that are located on chromosome 5DS, because both normal soft and SuperSoft wheat, they carry the wild type alleles at the hardness locus. To answer this question, we investigated the phenotypic variation of grain hardness in 172 advanced breeding lines and cultivars that are from the Washington State soft white winter wheat breeding program. And those lines are growing in multiple environments in Washington State in 5 locations and 5 years. The phenotyping for grain hardness was done using the single kernel characterization system that I described earlier, and the test was done on 200 kernels per sample. The genotyping was done using the genotyping by sequencing approach, And both phenotypic and genotypic data were used for association mapping to identify significant markers that are associated with grain hardness, and with particularly with the super soft kernel texture. The significant marker from the GWAS, or the association mapping, were converted into user-friendly KASP assay markers, competitive allele-specific PCR markers, that are now in the process of being validated on different genetic backgrounds so that they can be used in marker-assisted selection. Grain hardness in the Washington State University soft white winter wheat breeding lines and cultivar showed high heritability, which is an indication that the phenotypic variation in grain hardness is mainly due to genetic differences. But we also saw evidence of environmental effects. The grain hardness showed continuous distribution in this wheat population, and this is an indication of the involvement of multiple QTL associated with grain hardness. The super soft phenotype was observed in this wheat population, but in low frequency. Only 10 genotypes showed hardness indices of 12 or less. In this figure, I am showing the distribution of grain hardness in multiple environments for 15 commercial soft white winter wheat cultivars, where the box plots represent the different values of grain hardness in different environments. So we can see there is environmental effect within each variety. And we can see also this striking difference in terms of grain hardness between cultivars like Jasper, that is classified here as super soft wheat, where grain hardness can go up to -5 in some environments, while other varieties like Madsen, Serfa, and Pearl show the highest values in grain hardness, and they are classified as extra hard varieties. Association mapping was done using two models, mixed linear model and farm CPU. Both models, they identified a major locus on chromosome 5A. And the farm CPU was more powerful as it allowed the identification of additional associated markers on chromosome 2B, 3A, 3B, 6B, 7A, and an unaligned chromosome that were not identified using the simple mixed linear model. None of the association were found on chromosome 5DS, where we know that the pterandoline genes are located. So this is an evidence that the variation in grain hardness in this wheat population is independent of the pterandoline genes. In this table, I am showing the allelic effect of the significant markers from the association mapping on grain hardness. So here we have the 7 associated markers, and the green allele is associated with softer kernel texture. And for each marker, the wheat lines were divided into 3 classes. The wheat lines with the homozygote major allele, the heterozygote group, and the homozygote minor allele. And Tukey's test was done on grain hardness for these 3 classes, and this was done for each of the markers. The last column corresponds to the difference in grain hardness between the 2 homozygote groups, where higher value correspond to larger effect markers. This analysis allowed the identification of 2 markers with large effect, one on chromosome 3A at around 260 megabase pair, and a second marker on chromosome 5A that is located at around 664 megabase pair. And the difference in grain hardness due to this marker ranged between 12 and 13. This marker not only had larger effect, but also can distinguish between the super soft and the normal soft phenotype, as shown in these 2 examples of on chromosome 3A and 5A. And for the remaining markers, the effect were smaller, and the reduction in grain hardness indices were within the range of normal soft wheat. One of the largest effect marker on that we identified was on chromosome 5A, and this marker showed the clearest separation in between the super soft and the extra hard group, where all the lines in the super soft wheat class carry the T allele, where all the lines in the extra hard group, they carry the C allele. So this shows that this marker is tightly linked to the causal gene associated with the super soft phenotype. And this is an important marker that we are behind to breed for super soft kernel texture in wheat. In summary, super soft grain texture have the potential to improve wheat processing and flour quality. This phenotype is present in low frequency in the Washington State University soft white winter wheat breeding program. And the association mapping identified 7 loci. 2 of them had large effect and could distinguish between the super soft and the normal soft phenotype, while the remaining 5 loci had smaller effect and they could reduce grain hardness, but within the range of the normal soft wheat. The significant marker, SNP markers from the GWAS were converted into KASP assay markers. They, as shown in this figure, they can clearly distinguish between super soft cultivar like Jasper and an extra hard cultivar like Zerfa. And these KASP markers now are in the process of being validated on 2 biparental crosses so that they can be used in marker-assisted selection. With that, I come to the end of my talk, and I would like to thank the people who contributed to this work and the generous support from the USDA. Thank you.